Okay. Hi, everyone. Welcome to the Division of Rheumatology Grand Rounds. Thank you for joining us. I just want to remind a couple things that we will be using the Q&A box that you'll see at the bottom of your screen for all of the questions. And Dr. Knight will save about 10 to 15 minutes at the end of his lecture to answer those questions. And please note that there is a chat box as well as a Q&A box. Please put your questions in the Q&A box. And then Dr. Lude is going to help field those questions at the end of the lecture. And now I'm going to pass things off to Dr. Lude. Thank you.
Thank you. So it is my great honor to introduce Dr. Knight. He's an associate professor of rheumatology from University of Michigan. Dr. Knight obtained his PhD and MD degree at U Michigan and also completed his residency and fellowship over there. He has been very productive with over 66 publications, several of which have been published in top-tier journals, including A&R, JXMed, JCI, and also recently Nature Communications. He has been recognized for his extraordinary work and received several honors, including the Edmund L. Dubois Award for Young Lupus Researcher, as well as being awarded Marvin and Betty Dancer Research Professor of Connective Tissue Research. Other than being the Associate Director of the Lupus Program and caring for his patients and mentoring lots of trainees, Dr. Knight is also leading a very successful research program and has made many seminal discoveries, particularly pertaining to the role of platelets and neutrophils in thromboinflammation in lupus and APS. And more recently has also made several very interesting findings on neutrophil activation in COVID-19 patients, some of which he will share with us today. And as Lara mentioned, please send your questions via the Q&A function, and we'll try to address them towards the end of the presentation. With that, I will let Dr. Knight take over and share his intriguing findings with us.
Okay, thanks, Christian. This is super weird to be giving you a talk from Ann Arbor. This morning, my hound dug up a mole and apparently ate part of its head, but I have a great wife who, while I was seeing patients virtually, she took care of that. So basically, it's a Tuesday here in Ann Arbor. So let's start the way I start every talk, and this is a little ironic. Normally I try to bring this with gusto that we would not be a country without lupus and APS and how they got us here. Today, a little sadder to think about that, but if research can't bring us together for a common cause, then what can? So this is my traditional starting, and we're going to do it. So to think about how lupus and APS led to the United States of America, you have to think about the line of succession of the British monarchy. And so from Hamilton the musical, you're all pretty familiar with George III. He was perhaps not a great leader and certainly did not get along well with the American colonists. And you can contrast him with Queen Anne, who by all accounts was much beloved. Many things are named after her, including the Queen Anne style of house. And so it's my understanding that Queen Anne's Hill in Seattle was named from that type of house showing up there early on in the 1800s. By all accounts, Anne was a great unifier, and so you would contrast her with George III. England and Scotland came together under the flag of Great Britain for the first time during her reign. But so why this transition? Why transition from much beloved Anne to the House of Hanover and ultimately George III? Well, the problem is that Anne's health was not good. And so the historical record seems quite solid that she had a diagnosis of lupus. And so this is based on written records, but also portraits of her where she was noted to have malar rash and polyarthritis and edema. But I think also interesting to note that she had kind of this APS flavor of lupus with things like seizures and nosebleeds, potentially from thrombocytopenia, and she ultimately died of a stroke. But if that doesn't convince you, check out her pregnancy history. So, some 17 times she attempted to make an heir to the throne, and all 17 times was unsuccessful, just a disastrous history of stillbirth after stillbirth. And so that would be the argument. If not for APS, there would have been an heir to the throne, and we might have had someone on the throne who got along better with the colonists. So, makes me wonder, as I was born in Indiana, if I would have been born a British citizen instead of American. So Queen Anne's lupus is a way to think of APS. The last attempt to give this classification criteria was back in 2006. These are the so-called updated Sapporo criteria. There are a number of screwy lab tests that I'll try to give you a little bit of historical context for. I mean, for me, that helps me understand how we got here. And it's also a sad state because, you know, to be classified as APS, one of these really bad things needs to happen to you. This is not appealing to either patient or physician, obviously. But APS is best known for its predisposition towards thrombotic events, and it has this ability to trigger thrombosis in really any vascular bed, and also, of course, known for pregnancy morbidity and especially late pregnancy losses, you know, losses in the second and third trimester. As you probably know, this is a field that grew out of the field of lupus, and that's how we got some of the names like lupus anticoagulant, that this was first identified, you know, back in the 70s and early 80s in lupus patients. So why study APS? Well, you know, you are very educated as a rheumatologist, but I think it's more common than many people believe. You know, it technically meets criteria for rare disease falling under 200,000, but it's just below. I mean, it's almost as common as lupus, and this is based on epidemiology in both the USA and the United Kingdom. And then if you're writing a grant on this topic, you need to try to convince the reviewer that, you know, by studying APS, we may unlock secrets to very common conditions like stroke and pulmonary embolism. Now, you know, no one can argue that these are rare. These are major causes of morbidity. And I do think we have learned some things from APS that, you know, we may be able to apply to the general population. And we'll see if when we talk about COVID with the last 20 minutes of the talk, if I can convince you of that. So let's go through the history for 5 minutes and see how we got here. So this turns out to be a story that starts with syphilis. And so that was in the 40s. And so in the 40s, it was learned how to isolate cardiolipin. So this is a phospholipid that, you know, I'm sure Christian has taught you is especially enriched in mitochondria. And so heart is a very excellent source of cardiolipin. And it turns out that syphilis patients develop anticardiolipin antibodies. And so this was the first way that syphilis could be screened for. It was a major health problem of the time, and so this was rolled out to a great scale, especially as our troops are coming back from World War II. Now, by the 50s, you know, also the false positive syphilis test had been branded. It was recognized that while the sensitivity for this test pretty good, the specificity not so much. And so even in the 50s, it was recognized that the most common non-infectious cause of a false positive syphilis test was lupus. So super interesting. So, you know, maybe the very first signs of, you know, the recognition of APS was this recognition of anticardiolipin antibodies back in the early 50s. Now, meanwhile, if that story is not confusing enough, here's another one. There was this parallel story in which, you know, it was being identified that a subset of lupus patients had what they were branding as circulating anticoagulants. So these are patients who, when tested in vitro clotting assays, their clotting times were prolonged. So, you know, that's what we see if someone has hemophilia. If you're deficient in one of these clotting factors, you will have a prolonged clotting time. And so the prediction might have been for bleeding, but eventually people got brave enough to write up what the actual observation was, and that was for thrombosis. So this paradoxical effect. And so we now know that the issue is that these antibodies are mucking up the phospholipid layer, which is critical to these, you know, in vitro clotting assays. That's where the clotting factors come together to activate each other. The specialized studies like DRVVT have been, you know, constructed with relatively more limiting levels of phospholipid to make them more sensitive for picking up this effect. But very good work back in the 60s, people getting brave enough to kind of step away from you know, that initial common sense and, you know, observe the actual pathology. So despite anticoagulants, what we saw was thrombosis. And so then, you know, probably the most important paper that's ever been published in this area was by Hughes and colleagues, including Dr. Elkin back in the early 80s. And so in this seminal report, you know, they compared about 30 patients who had what was now branded as the lupus anticoagulant with 30 patients that did not. And so there was a tendency for the positive patients to also have anticardiolipin antibodies, and so that's worth noting. But the strong effect was for thrombosis, so much more thrombosis in the lupus anticoagulant positive patients than not. And then some pretty good trends for other things we associate with APS today, things like pregnancy loss and thrombocytopenia and CNS disease. So critically important work. So by the 80s, things starting to come together a little bit. So the lupus anticoagulant phenotype was pretty clearly associated with thrombosis. It was also recognized that positive lupus anticoagulant often tracked with these anti-cardiolipin antibodies. But as investigators, endeavored to study this topic in the laboratory and began to purify these antibodies, there was a problem. Like these antibodies were, beginning to be branded as antiphospholipid, and that's the terminology that we continue to use today. But when you use them in a purified system, they don't bind to phospholipids very well. You really need that serum present to facilitate binding. And so that raised the question of serum cofactors. So were we missing protein cofactors in the serum that were serving as a bridge between the phospholipid and the antibody? And so in 1993 groups, the Dutch and the Japanese and the Australian groups all identified this first cofactor, which we call beta-2 glycoprotein 1. It was originally identified as an apolipoprotein, and so its other name is apolipoprotein H. It's a very abundant protein in plasma. It's around the 20th most common protein in plasma, but its definitive function is still, I would argue, kind of unknown. There's certainly evidence that it can do some scavenging things, and maybe that is its ultimate role, but you can knock it out in mice and they don't get a phenotype. So still a little bit mysterious. But what we do know is when it binds to negatively charged phospholipids, it opens up and that reveals a cryptic epitope in domain 1. And so it seems especially these domain 1 antibodies that are pathogenic. So you can see here beta-2 kind of serving as this bridge between the phospholipid and the antibody itself. And so as we talk about some of the research we've done in our lab, we found that these anti-beta-2 antibodies are the ones that are very good at triggering netosis. And so there's a second cofactor, which I think a lot in the field are skeptical about, but I'm not sure. I think the main reason for that is that it has not been around from the beginning. I'm increasingly becoming convinced that we need to be studying this cofactor more, and that's prothrombin. And so you can look back to the mid-90s was when people really started to look at this, and the observation was that prothrombin antibodies are correlating well with lupus anticoagulants. And so it's known that anti-beta-2 antibodies, if you have high levels of them and purify those, you can cause a lupus anticoagulant phenotype to happen. But it seems a second cause of that, and some might argue the most important one, is these antibodies to prothrombin. And so I think the important, or kind of what brought us into the modern age and made this more practical was work from the Japanese group, including Dr. Atsumi. And so what they found is that prothrombin alone as an antigen did kind of okay. It had some sensitivity and a decent specificity for lupus anticoagulant, but they had better success if they first coated a plate with phosphatidylserine and then bound the prothrombin on top of that. I haven't seen anyone that has really explained exactly what's going on there in terms of specific epitopes, but presumably this somehow changes the way that prothrombin is presented to reveal its antigenicity better. And so this has been commercialized. I think the only available option to test this in the US is from Innova Diagnostics in San Diego, but they offer an anti-phosphatidylserine prothrombin antibody. I will say this is a test that we are sending, I think, more often nowadays at Michigan. We find it especially useful in patients who are on anticoagulation and we have some doubts about the trustworthiness of the lupus anticoagulant testing. And so if you have a patient who's negative for anti-beta-2, negative for anti-cardiolipin, you feel like you can't trust the lupus anticoagulant, I think this is where this testing can add value. And I feel like we have one patient who had catastrophic APS where we were having a hard time convincing pathology to plasmapherese them, but when we found high titers of these antibodies, that got us over the hump. And so I think that's an example of how I think this test has made some difference for us. And I partly introduce it here because when we talk about COVID, I'm going to need to remind you about it. So what can we say about pathophysiology of APS? Well, if you're confused by it, you can join me because I also find it confusing. I don't think anyone has put together, a complete explanation for what's going on. There's certainly a role for complement, especially in obstetric APS. I think Jane Salmon's work with patients and also in mouse models has been pretty convincing on this front. Investigators like Pierluigi Moroni and others have shown us that there's an important role for endothelium. And I think this is where the two-hit concept comes in, that you can have a sticky blood that's circulating, but in the end, all thrombosis is local. And so, something needed to happen to that particular endothelium to tip it from, an anticoagulant effect to a procoagulant effect. So monocytes have been looked at for their role in expressing tissue factor and production of prothrombotic microparticles, and also production of cytokines, including type 1 interferons. And then neutrophils, which is my favorite. And so, this is where we saw back, 6 years ago when we started to get into this area that it was understudied. And so we thought we might be able to bring some value in neutrophils. And so this is the main area that my lab has worked on. So let me tell you about that. So just to get us all on the same page, neutrophils, do not divide once they're in the circulation. They're terminally differentiated. They have this unique type of nucleus, which, I like to think helps them squeeze wherever they need to go, whether that's between endothelial cells or winding their way through tissues, like neutrophils get where they need to go. They're best known as the first responders. They're designed to have this innate immune response before the adaptive response has been fully set up. As a neutrophil researcher, I'm contractually obligated to remind you that the human body makes so many of these cells, like 200 trillion neutrophils produced every day. Wow. That's usually when I'm doing this live when the audience gasps, and I assume you did that at home. So circulating neutrophils last longer than we once thought, as labeling mechanisms have gotten better. The latest data would suggest about 5 days in circulation before they die by apoptosis. Shorter in mice it may be less than 24 hours. And then the other thing, as a rheumatologist looking at neutrophils, you have to use some type of terminology like double-edged sword. We all agree that neutrophils are important in protecting us from infection, but if not regulated properly, they have a lot of toxic stuff in them and can potentially cause problems. And of course, you guys know this as one of the leaders in this area. So this is the other slide that, so this one, unlike a gasp, this one usually gets dead silent. So I think this is the most clever thing I've ever done. And yet almost never does anyone laugh. But today I will also assume you're laughing at home. So my favorite thing that neutrophils do is this release of spider webs, this release of nets. And so nets were first described in 2004 by a German group. This is a paper that's been cited now thousands and thousands of times. As first described, nets were these webs that were built especially of chromatin, like their main scaffolding comes from that nuclear chromatin, but then also decorated with all kinds of stuff from neutrophil granules. They have abundant elastase, and we have a lot of good antibodies against elastase. And so when you see images of nets, it's common to stain them with an elastase antibody. And so it's that overlap of something like elastase and DNA and histones that really lets you call this a net. In recent years, it's certainly been recognized that mitochondria can become intermixed with nets, and mitochondria important for the release of nets in many physiologic contexts, and then can actually end up in the nets themselves, which can be a very immunostimulatory concept for the immune system, especially in the setting of lupus. So early work was done by the microbiologists, but then the vascular people started to get involved, and just numerous groups have shown that nets can be prothrombotic, and presumably there are multiple mechanisms at play. Histones, when they're outside of cells, are actually pretty toxic. They're very good at activating platelets and endothelial cells. Nets, of course, contain proteases, including elastase, which I just mentioned. And there's some stories about how these proteases may degrade antithrombotic factors like tissue factor pathway inhibitor. There's evidence that nets can activate both extrinsic and intrinsic pathways of coagulation. And then their scaffolding function is hard to ignore. Like if this thing is released intravascularly where it's not normally supposed to live, it looks a lot like fibrin. Even by electron microscopy, it can be difficult to tell if this is a net or if this is fibrin. So a structure that seems to be pretty good at triggering clotting. So this was our first foray, our first, I guess my first last author paper, wow, 5 years ago in 2015. And so this was work done by Shri Yalavarthi and Meng He in my lab. And so just some snippets from that first paper we showed. So these were patients with primary APS. So this was primary APS who came, into our clinic doing okay, typically on warfarin. These were not in the middle of a clotting event. And yet if we measured things like MPO DNA complexes, which is a marker of net remnants in the blood, that we saw higher levels of those in an APS patient who was just walking around minding their own business. We found that if we purified neutrophils from the blood of these patients, that they had a hypersensitivity to release nets. When a healthy neutrophil adheres down to a cover slip, it typically just sits there. But when these APS neutrophils get that stimulation of adherence, they're very likely to shoot out a net. And then we found that we could make a control neutrophil look like an APS neutrophil by mixing it with patient serum. So something in that patient serum, was changing the way the neutrophil behaved. So we went on to show that antibodies are certainly one of those factors, and I can't rule out that cytokines such as type 1 interferons might also play a role, but we know that antibodies will do it. So here we have purified antibodies from a bunch of individual APS patients, and many of them are pro-netotic. I'm not going to show you all the data in the interest of time, but I'm convinced that anti-beta-2 antibodies are the best subtype of antiphospholipid antibody for stimulating netosis. We certainly see that from a correlation perspective as well. So here we've measured these net remnants again in the blood of patients and correlated that with their anti-beta-2 levels, and we do see, a nice correlation. The patients with the highest levels of nets in their blood are the ones with the highest levels of anti-beta-2 antibodies. So we did a bunch of papers in mice where we made our own new model that we developed with our colleagues in vascular surgery. We were lucky to be at a place, Michigan, where there's been many years of research, especially in the area of venous thrombosis. They have models, all the way from mice up to baboons. We have focused on the mice, and I'll show you, the type of data we get with those models in a few slides. But what we found in these venous thrombosis experiments in mice is that if we depleted neutrophils, we could really reduce thrombosis. That if we gave the mice DNase, and so we can inject intravenously the same pulmozyme that is given to cystic fibrosis patients down their trachea. If we inject that intravenously, that's very good at dissolving nets and very good at reducing thrombosis. And then we also found that neutrophil endothelium interaction, not surprisingly, is important. So if we interfere with selectin pathways or integrin pathways, that's another way that we can reduce thrombosis in these models. You say, well, that's nice. So how are you going to kind of pivot to your human study? How are you going to inhibit neutrophils in humans? And so this is not so straightforward. There's not a lot of drugs branded as neutrophil inhibitors. I think we can all agree that even if we could deplete neutrophils, that's not going to be a credible solution. There's going to have to be some level of neutrophil activity that remains to keep us, safe, especially from, things like fungal infections. We have looked at other diseases to see if they have any clues. I already mentioned to you DNase and cystic fibrosis. I think that's, when they started using DNase in cystic fibrosis, they didn't know it was because of nets, but now, now that's been convincingly shown. I think the sickle cell field is another one that we have watched pretty closely. There is some good evidence that selectin inhibitors in that can reduce these painful bone infarcts that happen. And so maybe that's something we could look at down the road. But our approach has been to think about drugs that are maybe already out there that do have some anti-neutrophil properties despite never being branded that way. And so I'm going to tell you a story about one of those today. So let me set this up. Here is how we kind of found our way into it. And this is work that's especially been done by Ramadan Ali in the lab. And so it grew out of a project that we were doing with David Pinsky's group in cardiology. And he studies these ectoenzymes, especially ones called CD39 and CD73. And these are enzymes that live on the surface of cells and help to regulate these purine species, not intracellularly where we often think about them, but in the extracellular space. And so in the extracellular space, ATP is a classic example of a danger signal. Like there's so much, both ATP and ADP receptors on the cell surface that tends to do pro-inflammatory things. And you could contrast that with adenosine. So on balance, adenosine tends to be anti-inflammatory. So, these are enzymes that if you knock them out, you tend to see pro-inflammatory effects. And that's been shown in lots of different models. But relatively understudied in thrombosis. And so we were doing some thrombosis experiments with CD73 knockout mice. And so this is one of the models we do where we perform a laparotomy in the mouse, and we narrow the IVC just below the renal veins. And so if you do that in a healthy control mouse, and so then you let the mouse recover, and it will run around for 24 to 48 hours. And then at the end of that time, you can assess whether it has formed a thrombus or not. And so here you can see compared to wild type mice, CD73 knockout mice really get an accelerated thrombosis phenotype. And I'm not showing you the data, but we found that these mice also have very hyperactive neutrophils, at least in the context of thrombosis. So we felt neutrophils were part of the reason that these something about producing adenosine to quiet neutrophils was lost in these mice. So that led to us looking at adenosine and its receptors more carefully. I think unlike ATP receptors, where there's so many of them, there's only 4 known adenosine receptors. Depending on the cell type, they're expressed at different levels, not surprisingly, and they also have different affinities for adenosine. If you talk about neutrophils, the adenosine 2A receptor is the one that is most abundantly expressed. And so one thing these receptors do is converge on adenylate cyclase. And as you know, if you activate adenylate cyclase, it triggers the production of the quintessential second messenger in cells, cyclic AMP. So cyclic AMP is good at turning on kinases like protein kinase A. And on balance, this is a pathway that is anti-inflammatory. So if you activate these GS types of G protein coupled receptors that recognize adenosine, you are going to turn on cyclic AMP and you're going to reduce inflammation. And so we've tested this in vitro. So now instead of using adenosine, we're using specific agonists. There's been a lot of development in this area. There's a lot of good reagents out there that are quite specific. And so we show that if we activate adenosine 2A receptors, we are able to reduce netosis in vitro. So this is in vitro. And so yeah, adenosine 2A receptor, which is the most abundant on neutrophils, seem to be the most important one in our hands. So now we have tested this in mice. And so this is similar to what I showed you before, but now we're going to give the mice antiphospholipid antibodies that we have isolated from patients we see in the clinic. We, like others, have tended to use whole IgG fractions. If you take a patient who is triple positive at high titers and you purify their antibodies, their whole IgG fraction, and put that into mice, you do tip the balance toward thrombosis. And so we showed that if we also give those mice an adenosine 2A receptor agonist, that we are able to reduce thrombosis. So pretty nice. A question that we got as we were first, circulating this data was how do we know it's neutrophil specific? Certainly platelets and endothelium are also well known to have adenosine 2A receptors. And so we don't have this perfectly answered, but I think we're getting there. We have made the neutrophil specific adenosine 2A receptor knockout mice. And we have tested that in thrombosis models. This is a different model we've been using more recently, where we, instead of narrowing the IVC, which gives us so much kind of mouse-to-mouse variability, we are able to trigger local electrolysis by applying direct current to a copper wire that has been carefully inserted into the IVC. And that will locally activate endothelium and kind of provide a nidus where that thrombus can start. And so I'll be showing you more of this model later with some COVID context. But here what you can see is that when we have the mouse that lacks adenosine 2A receptor on its neutrophils, we've not manipulated these mice in any other way. We've not given them any extra stimulation. They're kind of naturally prothrombotic. And so I like to think of these adenosine 2A receptors as a natural brake on inflammation. And so the question would be, could we harness that to actually give a therapeutic effect in mouse or human? Yeah, at the same time of triggering more thrombosis, we also see that these mice are like hypernetotic. Their neutrophils are hyperprimed to release nets. So the only adenosine 2A receptor agonist that's branded as such that's currently approved for treatment in humans is a drug called Regadenoson, which is given for cardiac stress testing. There is another drug though called Dipyridamole, which I will tell you about now, which seems to harness this pathway, but just a little more indirectly. So before Regadenoson was available, IV dipyridamole was often given for this type of cardiac stress testing. If you acutely and in a large bolus hit coronary endothelial cells with an adenosine 2A receptor agonist, they're very good at dilating in response to that. If you administer a drug like this chronically, though, it seems the cells really quickly become potentiated to that effect. And so with chronic administration, you get antithrombotic properties. People have especially focused on platelets, and dipyridamole is typically thought of as an antiplatelet drug, but I'm going to show you that it also has some anti-neutrophil properties. So here's dipyridamole mechanism. So what it seems to be good at is blocking uptake of adenosine, and so it helps to make that halo of adenosine around the cell denser if you administer dipyridamole. So indirectly, pushes more signaling through adenosine 2A receptors, which will be anti-inflammatory. And so if we put dipyridamole onto neutrophils in vitro, so here we're measuring netosis, we're able to reduce it. And if we test it in vivo, it also works pretty nice for us. So, here's showing mice that have been treated with antiphospholipid antibodies. If we add dipyridamole on top of that, we're able to reduce thrombosis. And at the same time, we see lower levels of nets in the blood. Okay, so now, if you had asked me January 1st, we had been talking about we need to do, a single site dipyridamole trial for APS. We at least need to have biomarker endpoints and see if we can reduce some of these inflammatory markers that we see in their blood. However, then everything changed, as you well know. And so for you guys, it changed, I know, earlier than this, but this was the day that I started paying attention to COVID in a very intense way. And so I happened to do my 2 weeks of rounding in the hospital the first 2 weeks of March. Like, I've really kind of locked myself into this time. It seems the fellows are really optimally trained up at that point, but they're not kind of in senioritis mode. So it's a really good time I found to be rounding, but this year was not a normal year. So the first week of March, no problem. We, like all Americans, were ignoring this issue because it wasn't happening to us. But then the second week, everything changed. And so I think this was the second patient at our hospital that got diagnosed with COVID. We were called to see as the rheumatology consult team. This is a person who had not traveled out of Detroit. There was no reason to test them for COVID. It wasn't, on the radar. And so we were called to treat, what was thought to be lupus pneumonitis. But for us, this was very different than anything that had happened to the patient before. And we were worried something else was going on. This was still the days when I didn't know what we were allowed to write in the chart. COVID was being talked about in kind of whispered tones. So we wrote kind of this vague thing in the chart, but then stat paged ID to see the patient and let us know what they thought. This was also the era of 96 hours to get a COVID test result. So it was 4 days later before it was really confirmed that this patient had COVID. So that's when I got hyper interested. And I guess like everyone else before me did the same thing, right? So you see what has been published out of Wuhan, like what the heck are we supposed to do about this? And so what were preprints in February, I guess started to become papers in March. And so one of the things that, not something that we novelly noticed, but that a lot of people noticed, is that patients with severe COVID, the ones that did not survive, that had bad outcomes, had low lymphocyte counts, but at the same time had overall high white blood cell counts. So this is a paper that does not mention neutrophils, but we can assume that neutrophils are the drivers of this. And this is held up in many other cohorts, including ours at Michigan. So this is also a time when we start talking about cytokine storm, right? Like if my parents heard about cytokine storm before this, I think definitely not, but now, now they have. And so this is how rheumatologists, became somewhat on the front lines, because the drugs we knew about are now suddenly the drugs being used off-label in the hospital to try to keep COVID patients from progressing. So then March 15th, my time rounding ended, and it looks like I sent out this tweet wondering about NETs and coronavirus. There was nothing in the literature at that time that I could find, nor is there anything on medRxiv. Sending this tweet actually started a bunch of collaborations, and so we're now lucky to have a very good group of collaborators at the Northwell Health System in New York that we kind of identified through this accident of Twitter. But what was curious, we thought we've been interested in NETs and thrombosis. If there are more NETs here, then why aren't we seeing more blood clots? And so, if you looked for reports of blood clots in March, there was not so much, but we now know that that's different. I think May especially was the month when blood clots and COVID started getting all kinds of hype. So both in the popular press and then, especially in these autopsy studies. And I think interesting to see that in COVID, description of blood clots in really all vascular beds, arteries, veins, and the microvasculature. So that piqued our interest. So we set about looking for NETs in COVID-19, and this is the paper that we've published on this topic. We have a few other preprints up that I will show you here shortly, but this is the one that's been published. And I should really emphasize that, our efforts since April 1st in this area has really been a collaboration between Yogan Kanthi, who's a cardiologist at Michigan, and Ray Zhu, who's a rheumatologist, and then also me. Although I have not seen either of them in person in like 2 months, we have had so much conversations. And so this is the cohort we've been working with. At the time of the first paper, we only had 50 patients, but we were afraid for getting scooped, so we really rushed that out. But we've now analyzed 172. I think one thing about our cohort, we have communities like Ypsilanti and Detroit that have, are really enriched for Black and African Americans. And so a number of those patients have ended up at our hospitals. And so about half of our patients are Black. And then of the samples we've analyzed, more than a third have been on mechanical ventilation. A lot of samples we have are from March. And so we also have some less severe patients. That was a time when everyone with a COVID diagnosis was getting admitted to the hospital, even if they were on room air. I think that changed as time went along, and you really needed to have an oxygen requirement to end up in the hospital. But so we do have, even though these are all hospitalized patients, we do have a decent spread of severities that we can look at. The other thing to point out is that, we initially looked at how we were going to consent patients and get, really pristine research grade samples. And just asking that question in late March, it was a non-starter. There was not enough PPE to send a patient recruiter into rooms to make this happen. And so we pretty quickly decided, not to let the perfect keep us from doing something that might be good. And so we, made a partnership with the biochemistry lab where, once they were done with samples, and sometimes those samples set for some time, they may have set for up to 48 hours, the whole time at 4 degrees. But once they were done, they would release them to our lab, and then we would aliquot them and freeze them down. And so that's, the data we have is with that type of sample. And so if we look at different markers of NETs, so one is kind of cell-free DNA, or another is MPO DNA complexes, or another is free citrullinated histone H3. So for all those markers, we see kind of the median level in patients hitting around the top of our controls. So certainly there's COVID patients that do not have, tons of nets compared to healthy controls, but then, half to a third do really have that net signature. We did a lot of correlations with, the clinical labs you've heard of, like C-reactive protein or ferritin or LDH or D-dimer, and we do see pretty good correlation. The more inflamed patients, not surprisingly, had more nets. And we also saw correlation with respiratory status. So if you look at room air patients versus patients on positive pressure ventilation, the levels of nets are going to be higher. The other experiment we did for a week before we got clarification from our institutional biosafety committee that this really needed to be done in BSL-3 and not in kind of our BSL-2 plus setup. So this we did for a week and now have put on the back burner. Only recently did 2 of my postdocs get trained for BSL-3, and so just last week we started getting into the BSL-3 facility to move some of these experiments forward again. But what we did here was take patient serum and put it onto healthy neutrophils, and we saw that it worked pretty well. These are just 2 different, these were done independently, but 2 different netosis assays in which we saw enhanced netosis in the context of COVID serum. So something in that serum seemed to have pro-netosing properties. So this started as a side project, but then it worked so well, we really kind of expanded it out. As I mentioned, we had imperfect samples, and so we got good advice from Christian Lud to look at this biomarker called calprotectin. So this is the stuff that the GI doctors, measure in stool. This is a robust biomarker, and perhaps, we would get a better reflection of what was happening in that serum at the time that, it was drawn out of the patient's arm. And so calprotectin, as I'm sure is it has antimicrobial functions through its chelating properties. And why it works so well as a neutrophil biomarker are a few things. So it's definitely the most abundant protein in neutrophil cytoplasm. So neutrophils express a lot of this. And if they either die or get activated, that calprotectin goes from cytoplasm out into the world. It diffuses well from tissues to blood because it's a relatively small dimeric complex. And it's pretty good at resisting degradation. So all those things are good for its biomarker properties. When we look at 172 patients with COVID versus controls, we see they clearly have higher levels of calprotectin. But then it really worked better in some of the correlation studies than NETs did. So a nice correlation with C-reactive protein, pretty okay with D-dimer. But where it really stood out was when we were looking at respiratory status. So although it does not distinguish patients on room air from patients on nasal cannula oxygen, it does distinguish both of those groups from mechanical ventilation. So patients that are the sickest are also the ones that have the highest levels of calprotectin. And here's just another way of asking that same question. So here we're looking at the ratio of pulse oximetry to fraction of inspired oxygen. So a number like 500 is very good. That means you're at 100% oxygen and you're basically breathing room air. So like 0.2 for your oxygen. A number like 100 is very bad. That means you are, if you're satting well, it's on the maximum oxygen support. And so again, you see this negative correlation. So high calprotectin means bad oxygenation. And then what we thought was especially cool is you can kind of do it like a prediction style. So, here we limited our analysis to samples that we had from day 1 or 2 of hospitalization. And then we asked if patients ever ended up on positive pressure ventilation or not. And so you can see that there was a clear difference. So calprotectin level when you roll into the door of the hospital is pretty good at predicting whether you will end up on mechanical ventilation. So what about thrombosis? This is the area we're always most interested in. And so we've had a few different iterations of this. We saw people just publishing everything. So we thought we could write like a 3-patient case series and like send that to New England Journal, but I think we missed that window by a few weeks. And so we've eventually settled on what I think is a pretty cool case control study. So we have 11 patients from our cohort that had a thrombotic event, and we matched each of them to 3 controls matched by both age and sex. There was a few patients with stroke, but the majority had a venous thrombotic event. And I think worth noting that their ultimate outcomes were not different. So it wasn't just that the thrombosis patients had more severe disease. They were actually more likely to get discharged from the hospital than their matched controls. But if we look at nets, it was pretty clear. So also looking at calprotectin, so calprotectin and then the different markers of nets. And in all cases, we saw higher levels in the thrombosis patients as compared to the controls. So I think an argument for further investigation of whether nets are important drivers of thrombosis in COVID. And here's just another type of analysis where we compared calprotectin to D-dimer, also compared cell-free DNA to D-dimer, and see a pretty nice correlation suggesting that these activated neutrophils are correlating with the coagulation system hyperactivity of these patients. So we wanted someone else to do a dipyridamole clinical trial, but no one was interested in this. So we decided to do it ourselves, and this was especially spearheaded by Yogan Kanthi from cardiology, who I mentioned previously. And so our argument was that dipyridamole is good at increasing extracellular adenosine, and that might be a way that it could quiet neutrophils. And a second property that I didn't mention before is that it also has some activity as a phosphodiesterase inhibitor. So further potentiating that pathway, when cyclic AMP does get formed in the cell, it's less likely to be broken down in the presence of dipyridamole. And like, how might that work in COVID, where I think there's a lot of cell types that it could act on that would be productive, especially if you think about this thrombotic risk. People know dipyridamole is an antiplatelet drug, and so that's appealing to think about. The hope is that you would see less thrombotic events, but hopefully better disease overall. And I don't want to get too close to hydroxychloroquine. It's a little dangerous, but there are some papers showing dipyridamole has antiviral properties, including for COVID-19 specifically. Going back to the 80s, it has been shown to potentiate interferon responses, and that's one area that COVID patients, as you know, are felt to be defective. They don't trigger their interferon response as well as they could. And it appears that dipyridamole directly interacts with the virus's main protease, and so that could be another way that it would interfere with replication. So Yogan somehow figured out how to get the pharmacy to take 4 little tablets and put them into a capsule. So this let us make a placebo control. And so you can look at this on clinicaltrials.gov if you want to get more details. We're using the full dose of dipyridamole. We initially had some different tiers, but did not have the power we felt. And so ended up going for just placebo versus a full dose. I think the key to the inclusion criteria is that we are hoping to enroll patients who are requiring oxygen but are not yet on mechanical ventilation. Those patients who may progress over a few days of being admitted to the hospital to having severe COVID and requiring the vent. And to get the power calculations to work, we ultimately settled on D-dimer. And so that is actually the primary outcome, is can we affect how D-dimer changes during a hospitalization by whether someone is on dipyridamole or not. We certainly have a lot of secondary outcomes related to clinical status though. And so we felt pretty good. We had done a lot and it was time for a day off, but then everyone started writing these papers about antiphospholipid antibodies in COVID. And so first in New England Journal out of China, and then another, and then another, and then another. And so we felt that we were obligated as people of the antiphospholipid research to look into this in our cohort. And so this data is not published and is not yet on medRxiv, hopefully within a week or so. So I should point out that we tested 8 different antiphospholipid antibodies. So only 4 of those are technically part of the classification criteria. We also tested the IgA species, and we tested anti-PSPT, the one I introduced to you earlier. And so if you use factory cutoff for these ELISAs, half of patients, you can say they have a positive test. And if you go to a more stringent cutoff of greater than 40, which is frequently been used in the APS field, even then approaching a third have a positive. Most of those positives seem to live in the anti-cardiolipin IgM and then in the realm of anti-PSPT. So what about clinical correlations? So it's certainly not perfect, but there do seem to be some interesting findings. Anti-cardiolipin IgM was one of the most abundant, and it showed correlation with oxygenation and inflammatory markers and cell counts and also neutrophil activation. So that's interesting. And then if we look at PSPT, both IgG and IgM, some more hits and correlations there with PSPT IgM also correlating, albeit weakly, with oxygenation status. So it was not an easy thing, but we eventually got the approvals to do the experiment we're used to doing with APS. So what if we purify IgG fractions out of these COVID patients and inject those into mice? What is going to happen? And so that's what we've been working on over the last couple weeks. So we're using this electrolysis model of thrombosis, which gives us a relatively more consistent result. We can get away with using less mice. And so these are the data we have as of today, June 2nd. More experiments underway. First I would point you, so this is actually a pool of 2 patients that had catastrophic APS. We like to use these catastrophic APS samples because we have plasmapheresis bags. We have kind of unlimited sample, and so we can use them over and over again. And so this is what a CAPS IgG looks like. And then these are 3 patients that had high levels of PSPT IgG. And so they're showing signal actually stronger than CAPS. And then even some of the patients, our prediction was patients with lower levels of PSPT IgG, we would not see signal. And we are, it does look like we're seeing something there. So more to come on this, more samples to be tested, but could be quite intriguing. The problem with PSPT is that no one has ever done mechanistic work. Like, we just don't have an understanding. It's fine to say that this correlates with lupus anticoagulant, which it does, but like, what is it doing? What is its mechanism? There just is nothing in the literature to help us with that. And so one thing we've kind of stumbled into in the last few weeks, and so this was Ray Zhu's, one of his presentations at last year's ACR meeting. He has been interested in this anti-net activity in APS patients, and we've shown that some of these patients, because of these antibodies, are very poor at breaking down nets. And so we had never really looked at PSPT in this context, but both PSPT IgG and IgM are really correlating pretty good with those anti-net activities. And so, is high anti-PSPT IgG itself pathogenic, or is it a marker of some other activity in the blood that might be pathogenic? This is what we need to now figure out. Okay, this is the last slide until I get to my wrap up. And this is because I have a hypothesis that my spouse is tired of hearing about, but there's really no one else to tell it to. So I'm now going to tell it to you. And so this has been what I've thought since the beginning of COVID is going on. And we have yet to prove it, but we're working on that. And so as you know, COVID, or the SARS-CoV-2 virus, uses ACE2 as its at least most important receptor, it would seem. This is interesting to us because we've studied ectoenzymes in other contexts, as I showed you before, and it seems this is secretly, this pathway is thought of for its role in hypertension. Angiotensin 2 is very famous. Angiotensin 1 to 7 is probably one you've not heard of, but it has its own set of G protein coupled receptors and is actually has anti-inflammatory properties. And so our kind of pet hypothesis is that the virus is disrupting ACE2 function. Either the cells that express ACE2 are dying, or perhaps ACE2 gets internalized and its activity is just lost. And this is one of these natural breaks on inflammation. This is why things just go haywire. This natural break that's supposed to be present in the lungs is lost, and neutrophils and other cells go haywire. So things we're working on, we're expanding our APL testing. We are working on affinity purifying some of these APL from COVID serum. You know, the hope is to take what we learn about anti-PSPT, where we're having to figure out some stuff kind of from scratch. The hope will be to take that back to APS. And so, yeah, that's something I don't want to miss emphasizing. We have a really interesting project and a couple collaborations that are letting us do that, but to look at neutrophil-derived proteases. As you probably know, the spike protein, you know, needs to be proteolytically cleaved to be activated to facilitate membrane fusion. And so the question is whether any neutrophil proteases could be good at driving that. And then we're also looking into these different angiotensin peptides like angiotensin 1 and angiotensin 2 and angiotensin 1 to 7 and how they may regulate neutrophils. I should acknowledge, especially our patients. Yeah, it's sad actually. Especially now that we've gotten into COVID, this is the real deal. A lot of these patients have unfortunately succumbed to this, and you know, we're trying to do the best we can to make the maximum use out of their samples. Shri and Ray have really been working around the clock since April 1st. And then also just emphasizing collaborators, and especially Yoga and Kanthi, who you know, really has done half of everything that I showed you today. And then funding, just have to emphasize the internal funding that let us kind of move quickly into this area from the Frankel Cardiovascular Center and also the Taubman Institute. The Taubman Institute is sponsoring the dipyridamole clinical trial. We just recruited our first patient into the dipyridamole trial on Sunday morning. As these things go, it's the first one's always going to be on a Sunday morning, right? And you know, we'll see with as social distancing is lessened if we start to get more cases or not back in the hospital. So that's it. Sorry, I think I ran slightly long, but I started a little late because you guys were really like hammering one of your fellows, it sounds like, was giving a super long presentation. So, but now I'm done, and if we have a few minutes for questions, I can do that. I told Christian I was not going to look at the question box, but I would only do questions if he kind of fed them to me.
Wonderful. Thank you, Jason. This was really fascinating, I think. We do have one question in the chat, but I'll wait with that just for compliance. So I'll start with the one we have in the Q&A box. So this is from Keith Elkin asking that, I read that macrophages and neutrophils have ACE2 receptor on their surface. Could virus infection of these cells influence your result?
Yeah, that's a good question. So, you know, is there infectious virus in this blood or not? You know, people have certainly identified RNA in the blood at low levels, much lower than what you would find in like a, you know, in some type of respiratory swab. But, you know, you can find RNA there. But as far as we know, no one has shown infectious virus. And looking back to original SARS and looking at MERS, it seems the same. You can pick up RNA, but is there really infectious virus there? So to try to get ourselves out of BSL-3, we partnered with some other BSL-3 researchers who have a very sensitive virocell system for studying infection. And it does look like we find infectious virus in serum, at least at low levels. We are in the process of expanding that to see if we can sequence it and kind of prove it 100%. But I don't think it's impossible. It could be that virus in the blood, you know, in that, you know, one of those early experiments I showed you, that maybe was triggering NETosis. And if so, we need to figure that out. So this is something we're going to be working on in the coming weeks for sure. It's a good question.
Thinking about treatment, we have a question from Gordon Starkebaum asking whether methotrexate could increase adenosine and be useful to treat thrombosis.
Oh, wow. Yeah, I'd love that. Yeah, I love that idea. Yeah, this comes out of, I guess I give Bruce Kronstein credit for sorting a lot of that out, but I'm sure others also played a role. In that Nature Communications paper, you know, we tried to write a little blurb about methotrexate, but the reviewer made us take it out. They were not convinced in that mechanism. But there's the idea that methotrexate kind of mucks up purine signaling and forces different purine species out of the cell where they can get converted into adenosine. And so, you know, some argue that that's the primary anti-inflammatory property of low-dose methotrexate, the way we use it in rheumatology. So yes, I do not think it is a bad idea. I mean, what I don't know about methotrexate is how quick its mechanism of action is. Like, could you credibly give a dose of methotrexate on day 1 of hospitalization? Do you have to build up to that anti-inflammatory effect over a few weeks, or do you have it straight away? So we've gone with dipyridamole, but I think it's a good idea for sure.
Excellent. A few more quick questions here. This one from Thomas Mustelin asking whether there are any evidence that neutrophils play an antiviral role in COVID.
Yeah. Unknown, I would say, but I think it's an important point. Always the double-edged sword has to be commented on. I mean, maybe neutrophils are high in these severe patients because they have a lot of virus and they need more neutrophils to counterbalance that. Hopefully, as animal models come online this summer, as people grow up, the humanized ACE2 receptor mice, that these questions that may be very difficult to answer observationally can be dissected experimentally. But yeah, it's an important point that should be emphasized. I like it.
This one is from Slajana Gardner, and I think I can also expand on that, asking whether you think neutrophil activation happens first and then that you develop anti-net antibodies or antiphospholipid antibodies, or the other way around. Do you see any connection between antibodies towards the virus and anti-net antibodies?
Yeah, I think there's a lot to figure out. I mean, we've been thinking about it a lot and don't have it determined. I mean, I think it's, I guess as you guys know, antiphospholipid antibodies in the setting of infection, I mean, many a paper has been written about this. It's known to happen that these antibodies can be triggered transiently. But I guess I would argue that just because it's transient doesn't mean it's not causing a problem when it's actually there. And I think there's just very few studies that have looked at that at all. I think we may have a rare opportunity because we suddenly got 200 patients all coming at once where we can, try to figure some things out. And, it could have implications for, monitoring these antibodies in other settings. One wonders about convalescent plasma, things like that. Do we need to be, we don't have any testing from patients that have resolved and have left the hospital. But I think getting a very extended time course where we can follow patients from, the beginning of their infection until resolution and see what these antibodies do. I mean, maybe they're going to cross paths with the protective COVID antibodies. And by the time those arrive, these have faded away. But I think you're asking the question because there's a lot of important implications clinically, and I totally agree with that.
Excellent. And then one final brief question here from Natalia Giltiai, asking about what would be the component in serum driving netosis, if there are any specific cytokines do you think would do that, or whether it could be immune complex driven?
Yeah, we don't know. We don't know. I think, Dr. Elkin's idea for virus is important and should be, ruled in or out. I mean, lots of cytokines, IL-1, there's, IL-1 has been detected both upstream and downstream of netosis, and could it be playing a role here? We really see a tight correlation between platelet levels and, I mean, that wouldn't explain our serum results, but, in vivo, I wonder if neutrophil platelet, like in our more severe patients, we see higher platelet levels. That's what we've seen in our cohort. And one wonders about neutrophil platelet interactions and how that might be an important contributor in vivo. So I think, yeah, these are good ideas and questions that we don't have the answer to yet.
Wonderful. Thank you so much, Jason, for a wonderful presentation. And I'm sure that if there are any more questions, they could probably email you or find you on
email me or find me on Twitter. I love how you guys have set up that like parody room cat account. I've been a big fan of that. I assume you have like a team of comedy writers working behind the scenes on that one. So keep it going. I'm sure it's been a great recruitment tool for you. So I just wanted to shout out the room cat account.
Thank you so much, Dr. Knight. And thank you all for joining us. We'll be posting this on our website shortly. Okay, goodbye.